GemIdent was originally designed for cell phenotype identification in the context of a tissue sample. In this demo, we will process a tissue scan created by the Bacus Labs Bliss automated microscopy system. I'll create a new project in the directory of the scan files. GemIdent is well integrated with the Bliss image scan file format. I'll now initiate the training helper, which will display a thumbnail view of the entire scan. This tissue is an immunohistochemically stained lymph node from a patient with breast cancer, and it was imaged at 20x. The blue is a background counterstain for all nuclei. The red is fast red on AE1, AE3, which is a stain for breast cancer. And the brown is DAB on CD1A, and it looks for dendritic cells. In this demo, we will be attempting to identify only the cancer nuclei. We can load the Bliss subimages by clicking anywhere. Let's start with color training. We first load a representative image. We observe that there are 3 relevant colors: red from the cancer membrane, blue for the counterstain nuclei, and the third is the background white and the bluish-grayish intercellular matrix. We'll call this white. Let's give GemIdent 10 examples of each color. That should be sufficient. Let's start with red. I'm going to change the display color to white so I can see my training points with good contrast. Notice that as I train, GemIdent keeps a running representative sample of the color. If I make a mistake, I see the image become heterogeneous. I can delete the wrong point by using spacebar to toggle between mark and delete. The training points must be exact, so the use of the magnifier window on the right is encouraged. I can increase or decrease the zoom by pressing the plus or minus buttons. Let's finish up this training. Let's also change the display color of blue with something with a little bit more contrast. Let's be sure to include some examples from the intercellular matrix as well. Now, we need to hardwire the color information into GemIdent. We do this by pressing the compute button for each color. The reason for this deals with the internals of the GemIdent artificial intelligence engine. We'll let these compute and move on to the more interesting phenotype training step. Again, the one and only phenotype is the cancer nucleus. I will name it here by calling it CanCen for short. Let's color the training points neon green for contrast. Now let's load an image with lots of cancer and begin training. I'm trying to click the centers as best as I can. We must also give GemIdent an idea of how large the cancer nucleus is. We can do that by adjusting the Rmax value. Notice the helper display image gives small examples. I think a size of 13 is about right. Note that sometimes the presence of a nucleus is ambiguous. It helps to consult with tissue experts while training. Let's load another example image for a more diverse sample. I think about 30 various examples are enough. Now remember from the previous demo that GemIdent also requires knowledge of what the opposite of a cancer nucleus looks like. This is the non. Let's begin by giving examples of the membrane. Now the background with the non-specifically stained nuclei. Now I should give it edge examples so GemIdent learns not to include the edges of cancer nuclei, and it learns to separate nuclei from each other when they're closely clustered. Notice when I place a dot, GemIdent includes many nearby pixels. This is optimal for beefing up the dataset, but gets in the way of this sensitive edge training. Let's reduce the number of pixels to a radius of 1 by adjusting the RMIN. And we'll continue training. I think 100 or so nons is appropriate. We're now ready to classify the images. Let's classify the 2 trained, and also give it 10 random ones. GemIdent is currently building the training data and the machine learning classifiers. It is now complete and is currently classifying the 2 images plus the 10 random ones. This demo is being run on a quad-core AMD 2.6 GHz with 8 GB RAM running on Windows XP 64. GemIdent was designed with multi-threading in mind. With 4 cores, it will identify 4 images simultaneously. This is why we constantly observe the images switching back and forth as the identification moves in real time from the top to the bottom. The pixels that thinks belong to a cancer nuclei will be colored by our display color, the neon green. It seems to take about 34 seconds to complete each image, so there'll be about another minute or so left. Let's go over the classification parameters. The accuracy level refers to GemIdent internals about the machine learning classifier. For those interested, GemIdent employs random forests, and the accuracy level is the number of decision trees grown inside the forest. The number of CPUs is defaulted to the number of cores the operating system detects. One may wish to use fewer if other applications are demanding. The pixel skip, which is here, which is also 0, tells GemIdent to classify every single pixel, not skipping any. If the phenotypes are large, you may get an accurate analysis by skipping every other pixel, pixel skip of 1, or every 2 pixels, pixel skip of 2. This depends on your application. All this information is in the easily accessible manual. The Find Centers button will do blob analysis to try and locate the centers of the cancer nuclei. This feature will be demonstrated shortly. Let's wait for the identification to complete. It is now done. Of course, GemIdent made mistakes, so let's return to training and retrain over those mistakes. Let's begin with the initially trained images. GemIdent gives us these handy sliders that control the opacity of the classified pixels. We can also turn the visibility of the training points on and off. Notice the regions of false positives. Let's retrain over them with the none. We can also retrain over false negatives with the CanSen phenotype. Now we will boost the training set by looking over those 10 random images. Locating mistake in training will automatically add the image to the training set. See how the image popped up down here on the thumbnail pane? Let's go through them, correcting the errors. It's now time to reclassify. This time we'll allow GemIdent to post-process and find the centers as well. To save time, let's classify only those trained. Notice how GemIdent identification takes a toll on the computer itself. Identification is done. It will now try to locate the centroids. It will then give us a count and a false negative rate. That's pretty good. To make the identification even more solid, let's return to phenotype training. Here are some errors. You get the idea. A good classification may take many cycles of training, classifying, retraining, reclassifying. It all depends on the application and the accuracy desired. When confident, a full classification of all images in the set can take place. In the next demo, we will show another histological sample, with a full classification for many phenotypes already completed. We'll then dive into GemIdent's analysis features.